Hello there! Would you like to see what I'm working on? I'm currently analyzing intervoxel phase shifts caused by magnetically susceptible particles in rectus femoris tissue samples. Do you know anything about Laplacian-based phase unwrapping? You're probably confused. Let's start from the top.
Just like a phone can send and receive signals from cell phone towers, a radio frequency coil can send and receive signals from atoms. Atoms have an intrinsic property called spin. No, not that type of spin. No, not that spin either. Nuclear spin. But this is easier to visualize, so we'll stick with it. Atoms with a non-zero spin have a frequency at which they resonate. This frequency is unique for each atom, and it varies depending on the strength of the magnetic field. In a powerful magnetic field, the spins of atoms will align. We can then send in a pulse of RF energy at the resonant frequency for a nuclei of interest, which causes them to excite and tip in the direction of the energy. Eventually, they relax back, and as they do so, they release energy. This energy is picked up by the RF coil and converted to an image. Clinical MRI scanners only look at hydrogen, as it is by far the most abundant atom in our body. However, as this is the case, minute changes caused by physiological processes cannot often be measured, thus restricting hydrogen MRI to just structural images. Other nuclei that are important to the human body, such as sodium, phosphorus, and carbon-13, are less prevalent, and as such, pathological changes are easier to detect. A separate property that influences how atoms interact with magnetic fields is magnetic susceptibility. Here is a spectrum of common MRI-compatible molecules, along with their susceptibility values. And here is where they fit into the entire susceptibility spectrum. So while something like magnesium or copper may not be as magnetic as something like iron or steel, they are still definitely magnetic. All you need is a strong enough magnet to see that. While we clearly don't want patients floating in MRI scanners like this frog is, we can still use this phenomenon to detect molecules not visible with typical MRI, such as calcifications or blood iron. Consider calcium carbonate, a weakly diamagnetic molecule. When a magnetic field passes by the molecule, it is slightly repelled. And if you look at a magnetic field without the molecule, it goes straight past like you normally would expect. However, there's an interesting effect that you might not have noticed. If you look at both of them side by side, you can now see that the one that has passed by the calcium carbonate has an added phase shift. This is incredibly helpful as we can quantify and image this phase shift to extract helpful information in the resulting images. The most well-known use of this phenomenon is blood oxygen level dependent or BOLD contrast, which is used in functional MRI. This allows you to identify changes in blood flow in the brain. Another up-and-coming technique is quantum susceptibility mapping, or QSM. This enhances the phase shifts caused by magnetically susceptible particles, allowing you to see and differentiate between diamagnetic and paramagnetic particles, as indicated by the white and black arrows in the image, respectively. My research involves using QSM to study Duchenne muscular dystrophy, or DMD. This muscle disorder, characterized by a slow degradation of muscle tissue over time, is very poorly understood. And while only 1 in 4,000 males will be diagnosed with DMD, most of them will not survive their 20s. Typically, diagnosis does not occur until after onset of physical symptoms, thus preventing the study of the disease's early stages. However, post-mortem analysis of DMD animal models has revealed calcium deposits in diseased tissue, and it is believed that calcium plays a crucial role in the destruction of mitochondria and eventual necrosis of muscle cells. As such, I aim to use QSM to identify calcium deposits in diseased tissue samples to see if it could serve as an early biomarker of DMD.
So, would you like to help me? Or perhaps you could take over the project entirely?